Today I will talk a bit about our research at the, let's say, single molecule scale in order to unravel biomolecular processes in nature. I have only 10 minutes. I will try to not be cut and not to be long. So I would like just to give you an overview of what we do, but please don't hesitate to ask more specific questions. So why we need the single molecule studies? Mostly because it's very important to be able to understand nature. And when first nanotechnology has started and excited Feynman started, now it's very easy to solve any question in biology. It's simply easy to look at the thing, where the thing are the single biomolecules which are constituting life. However, the picture is a bit more difficult, and we need to consider also that these biomolecules have chemical properties, and these chemical properties are intrinsically related to their structure, which we normally measure by AFM, as well as their function. Then, as we know, and this is why mostly we use atomic force microscopy, we need to characterize these biomolecules at the nanoscale, because these are the intrinsic dimensions. However, while we normally are able by atomic force microscopy to characterize these species in morphology, is also important to be able to unravel these relationship between their chemistry and structural properties. One example of why this is important is the example of neurodegeneration. What we know that when we get Parkinson's or Alzheimer, one of the proteins that is normally present in our brain changes its behavior and start to aggregate into fibrils that we call amyloid. So we have indeed a morphological and structural change. However, what we also know is that this aggregation is triggered by a misfolding, so a conformational change of these proteins that leads to this aggregation into fibrils, that they have a universal structure, which is called cross beta sheet structure, where a lot of beta sheets are perpendicular to the main axis of symmetry of the fibrils, and the fibrils have only a diameter of about 5 to 10 nanometers. So it's really important to capture this correlation. And so what we do in our laboratory, we try to use methods based on scanning probe microscopy and spectroscopy in combination with microfluidics and sensors in order to unravel this process of protein self-assembly. But as well as taking inspiration by this process, we try to tailor the future class of biomaterials which could substitute plastic and so help us to overcome fossil fuel-based society. Today, I will focus mostly on how we try to develop this technology and how we try to bring nanoscale imaging just beyond seeing. So what we have to think is that if we have a nice beer, as we saw yesterday in the very beautiful testing, we see a kit, we see it's cold, it's summer, we want to try it, because seeing is not always sufficient to understand about something. So we smell it, we taste it, and this comes in a sort of chemical recognition that we need to understand the nature. And what I will show today is how we can use this to unravel properties of biomolecules at the nanoscale. An example that I can give more at the biomolecules levels is the following mental experiment. If we want to look at some protein fibrils as the one I showed you before, or nucleic acids at the nanoscale, we can put them in test tube. We spot on our surface and we can get beautiful images or force spectroscopies as we have seen in our conference in these 3 days. However, if now we take them and we mix them together and we spot them on our surface, now it's much more difficult to say what is what. And this is because we are chemically blind. A solution that science has taken up in the last centuries is trying to use bulk technologies and chemical approaches to differentiate what is what in our solution. And for instance, this is the case of infrared light. And by looking at a vibrational signature of the molecules, we can say that we have both the amide band of proteins as well as the phosphate groups of nucleic acids. However, these techniques are chemically especially blind, so we cannot have maps. So we cannot say exactly what is what. The solution comes in the combinations of these two words, so the physical and the chemical world, in what we call infrared nanospectroscopy. In infrared nanospectroscopy, what we can do, we can simultaneously have maps of morphology and maps of chemistry. Then we can point our tip on the top of each specific species in our surface, and we can acquire this vibrational spectra that enables to taste what is what in our map, and so going just beyond conventional imaging. This is very useful in the case of proteins, as is in the case of neurodegeneration, because by looking at the amide band 1 of these proteins, it's possible to understand their secondary structure, and so to understand this conformational misfolding that occurs in disease. But how the technique works? The technique works very simply based on a thermomechanical detection. If IR light is sent to our sample while we are scanning it, if the chemical bonds absorb the lights, will start to vibrate. And this vibration is dissipated as heat inside our material, and this thermal expansion can be measured by our cantilever. That is a very sensitive force sensor. So if we do this at every point, we have a chemical map. And of course, we have a force sensor, so we can get simultaneously also mechanical maps. Then if we point at a specific location in our map, we can get, by sweeping the laser, IR spectra at the nanoscale. And so we can have simultaneously the possibility to test, to see, to touch, and to taste our sample, similarly as we will do with a nice beer. Now I have very short time. I will not go too much in details, but I would like to show which are the latest breakthrough that we could bring in this AFM beyond imaging. We have started a few years ago where we first demonstrated that this nanoscale chemical approach could differentiate the structure of proteins at a very large scale, at the droplet scale. Then we have integrated the technique with microfluidics in order to study single molecules. We have shown for the first time that is very relevant for biomolecules that we can have liquid operation, as well as very recently we demonstrated that we can acquire IR spectra down to a single protein, still a large one. We are not really at the single molecule scale, but this was already an important achievement for the field. And these are the two last achievements I would like to give you briefly an overlook. So the first one is the possibility to measure in liquid. Similarly as we do in conventional ATR spectroscopy, we have a prism. The laser is now in total internal reflection, so we can curb the IR absorption of water. And what we could demonstrate is that we can acquire chemical maps in air as well as in liquid. And then we can point specific positions in our map, and indeed we can acquire IR spectra in liquid environment at the nanoscale, and we were able to differentiate the secondary structure of different protein aggregates. The second achievement I just showed you is the possibility to acquire spectra at the single molecule scale, towards at least. So what we can do, we can take a single protein, we can map them, we can map their morphology, in this case it was stereoglobulin, and we can acquire simultaneously IR maps. Then we can acquire IR spectra on a single protein in a similar manner as done by FTIR on billions of molecules. And by comparing their amide band 1 secondary structure, we can see that we can determine secondary structure of single proteins at the nanoscale. All of these achievements were useful in our studies in order to unravel this problem of protein misfolding and aggregation, as well as the problem of liquid-liquid phase separation, that is the precursor of this aggregation, as well as we could prove that we could study living organisms and how they can use these protein aggregates functionally. And as well, very recently, we could prove that we can target single protein interaction with drugs. So what we would call this field that we try to open up now is a field of nanoscale drug discovery, where we want to target the interaction between these amyloids involved in the onset of Alzheimer's with drugs, which can delay the onset of the disease in animal models. So we want to understand how this protein interacts with these single small molecules. And of course, what we can do, we can acquire IR spectra of each individual species, and then we can compare the spectra of the proteins alone with the spectra of the protein incubated with a small molecule. We can see that the spectra are different, indeed, in the IR absorption of the small molecule that delay the disease. And then what we can do, we can apply artificial intelligence and multivariate data analysis to see where the difference lay. And what we could demonstrate indeed is that the molecule is shifting its conformation when it's in presence of the aggregates and is binding to them. I'm sorry if I skip a bit of details, but I have very short time. it can bind to them through a single hydrogen bonding, thus proving that we can study how these species could be modified and their toxicity could be suppressed in vitro, but hopefully also in vivo to tackle these diseases. Finally, with this, I will be done, almost on time. And I hope I demonstrated that it's helpful to go beyond simple imaging, simple force spectroscopy, and that is useful to correlate the chemical and structural properties of biomolecules at the nanoscale to understand the rules of life. With this, I would like to thank you and to see you at the QI session. Thank you very much.